Hello there, it's Luke, and this is our second tutorial looking at Starling forces and tissue fluid. And in this tutorial, we're going to look at applying our knowledge of the Starling forces to different clinical situations, different pathologies, including dehydration, malnutrition, different types of edema, and also thinking about heart failure and fluid overload. So we'll start off with just recapping what we were talking about in our previous tutorial. Okay, so in health, there's a balance between these two pressures. There's the hydrostatic pressure, which is pushing or forcing water or fluid out of the intravascular space, so out of the capillaries and into the interstitium. And then the sort of big molecules within the capillaries, so things like proteins, albumin, big cells, that kind of thing, will be exerting a sort of osmotic pull, so what we refer to as oncotic pressure, drawing the fluid back into the capillary from the interstitial space. And as we mentioned before, in health, running alongside the circulation of blood, so the systemic circulation, we have something called lymphatic capillaries, and they're also permeable to water, and what they do is they help drain away that excess fluid that's been pushed out of the capillaries due to hydrostatic pressure but hasn't been drawn back in by the oncotic pressure. And for completeness, the excess fluid that is pushed out by the hydrostatic pressure but doesn't return back to the intravascular space we would refer to as net filtration. So hydrostatic pressure, oncotic pressure, net filtration, they're the three key ideas. Okay, so we're going to think about our first clinical situation. We're going to imagine a patient who is dehydrated. Okay, they have maybe they've been taking in less fluids, maybe they've had diarrhea, maybe they've bled, whatever the situation is, or sweated or passed too much urine, or whatever the thing is. And so they have less preload, there's less blood returning to the heart, their blood pressure you would think would be less, and their cardiac output would also be less. And so in our sort of healthy person at the arterial end, which is towards the sort of left-hand side of our diagram here, would have a higher hydrostatic pressure than at the venous end. However, in somebody who is dehydrated, you might expect that hydrostatic pressure to be less because there's less return to the heart, there's less preload, there's less cardiac output, and the blood pressure is likely to be less as well. And so it follows that the hydrostatic pressure, so that pressure that you principally get from the arterial blood pressure, will be less. However, there is no change to the movement of big proteins and cells from within the intravascular space, and so they're still exerting their oncotic pressure, so they're still drawing fluid back into the blood vessel. And so the hydrostatic pressure is less, the oncotic pressure is the same, presumably as in health, and so we have a net movement of fluid from the interstitial space, so from the space surrounding the blood vessel into the intravascular space, so back into the capillary. And thinking about how this might look in a patient, this might be exhibited as reduced skin turgor, by which I mean if you have a healthy person and you squeeze the skin and let go, it very, very quickly, because I'm well hydrated, sort of disappears, that area of skin that I've pinched. But in a dehydrated person, such as in this photograph, there's less fluid within the tissues, and so you might get that sort of skin staying put. As I say, we refer to that as reduced skin turgor. Now we'll consider a second situation whereby there are fewer proteins within the intravascular space, and this might come about in somebody who has a liver that is working inefficiently or is failing, or in a situation where somebody is malnourished, so they're not able to get in enough fuel and eat enough protein and all the things that are used to synthesize proteins. And so there are fewer proteins and big molecules floating around in the blood. Well, this will, as you've probably already guessed, reduce the oncotic pressure. If there are fewer big molecules within the intravascular space, there'll be less oncotic pressure drawing water into the intravascular space. However, this person may be well hydrated, perhaps, and so the hydrostatic pressure remains normal, and so we have far greater fluid leaving the intravascular space into the interstitial space than is returned. So there's a far greater net filtration, and we would recognise this clinically as edema. So there's an excess of tissue fluid, and we would recognise this as edema, and this is one of the causes for what we would refer to as peripheral edema. And you can see in this photograph, this is actually very, very severe edema when that person is pressing, and I think this would be quite a painful thing to do as well, pressing on the skin. It's actually leaving behind a mark because the tissues are so waterlogged and so full of that excess fluid. Now, as we said before, quite a few times in the previous tutorial, that the movement of blood through the capillary occurs due to there being higher pressure on the arterial side than there is on the venous side. But what happens if there's an increase in pressure on the venous side? So, and this could cause for this could be due to a couple of reasons. So, for instance, if you have a failure of the right ventricle, so that's the sort of portion of the heart that squeezes the blood that returns from around the body through the lungs, okay, out of the sort of venous return, and forces the blood into the lungs, if you like. So if you've got a failing right heart, then you might get a backup of pressure in the venous side of the circulation. Well, in health, or in a normal sort of well-beating heart, if you like, you can see sort of the hydrostatic pressure diminishes as it moves along. You go from the arterial side to the venous side of the capillary. However, if you've got an increase in pressure at the venous end, perhaps because your right ventricle isn't pumping effectively, then that hydrostatic pressure may be sort of maintained and elevated across the length of the capillary. Now again, there may well be the case that this person has normal amount of proteins or cells within the intravascular space, and so the oncotic pressure is normal, and so you have hydrostatic pressure being maintained along the length of the capillary, and it being higher than normal, but you have a normal oncotic pressure drawing fluid back in. And so again, you can see you've got this disparity between the two things, and so that you'll get a higher net filtration and you'll get edema formation. And again, we're in the situation where, in this particular person, that the edema is gathered sort of down in the sort of lower limbs. But of course, it can occur in other parts of the body, so it can occur around the sacrum, for instance, or actually it can occur within the abdomen as ascites, for example. Another example of this same phenomenon is if you have a blockage at the venous side of the circulation. That could be within the capillary, or more likely within a vein that the capillary would drain into. And so that blockage causes an increase in pressure along the length of the capillary, or along the length of the vein if we're further along than that. And so correspondingly, because that hydrostatic pressure is maintained along the length of the capillary, more fluid is being forced out. And if this is within a vein, perhaps within the leg, this might be referred to as a deep vein thrombosis, and this is why somebody who has a DVT, for instance, in the sort of right lower limb, has this sort of edematous, painful, swollen leg. Is because the vein is obstructed, and that has a sort of back pressure and a build-up of the hydrostatic pressure within the capillaries, and so there's a greater net filtration as more fluid moves out of the capillaries and isn't reabsorbed back into systemic circulation. Now, as you'll no doubt be aware, those aren't the only reasons why you may get edema formation. Of course, in both sepsis and anaphylaxis, you can have edema. You can have that sort of greater net filtration, that greater amount of fluid in the tissues. But in this instance, it's due to greater capillary permeability. So as we said before, a healthy capillary will let through some water or fluid, and there's that movement between those two fluid compartments. In sepsis and anaphylaxis, there's increased capillary permeability, and this is due to the action of inflammatory mediators, by which I mean things like histamine, which is particularly released during an anaphylactic type of reaction from the mast cells, but also inflammatory mediators such as bradykinins and tissue necrosis factors can also act upon the cells within the capillary wall themselves and cause them to sort of move apart, and so make the capillary more permeable. So there's bigger spaces through which the fluid can flow out. So to go back to our diagram with all the arrows, it's not that there's a greater hydrostatic pressure all of a sudden, it's just that there's much larger holes through which the fluid can leave the blood vessel and enter the interstitium. Now, this is complicated by a second factor. So one of the things that occurs during the inflammatory response is that you want white cells and proteins and all those helpful things to be able to get to the tissue to, say, fight the infection. And that's part of the reason why the capillary becomes more permeable, is to get those proteins out. However, as you've no doubt already worked out, if those proteins are then outside of the capillary and in the interstitial space, they too will have a oncotic pull or osmotic pull of fluid from within the blood vessel into the interstitial space. And so during sepsis in particular, you've got increased capillary permeability, and so hydrostatic pressure has more influence because there's a greater hole through which the fluid can leak. And then you've also got proteins within the interstitium which are keeping the fluid, exerting an oncotic pull, keeping the fluid within the tissue. And so you very sort of rapidly can develop edema. And this obviously will have consequence for when we start giving fluid boluses or giving IV fluid to these patients who are septic. You very rapidly will get edema within the tissues. And these slides just say the same. So the same things. And because you've got a loss of fluid from the intravascular space, you have decreased venous return to the heart, decreased preload, and of course a lower blood pressure. And then following on from that, perhaps failure of perfusion of the tissues. And here we are, we've got edema again due